Hello, everyone, and thank you for joining us for today's webinar entitled Increase Your Probability of Success with Faster, Easier Protein Expression in E. coli. My name is Karen Clayman, and I'll be moderating the event today. I'm very pleased to introduce today's speaker, Beth Fry, Product Manager here at Lucigen. Beth?
Hello, and thank you, Karen, and thanks to all of you for joining us today. I've been looking forward to talking with you today about one of our favorite subjects here at Lucigen, which is protein expression. And we express and purify many of our own proteins here in bacterial systems. And so we are familiar with many of the challenges that you may be facing with your own experiments. So today, what we're going to talk about is some of those challenges, some of those common problems, and how to mitigate them. And along the way, we'll work through some examples, and we'll also talk about a common challenge that some of our customers and collaborators have, which is scalability. So scaling these protein expression experiments, the cloning and the protein expressions for processing many samples at once. And then we'll finish up by talking about proteases that can be used for fusion tag removal and best tips and practices for using them effectively. So let's jump right in by looking at a fairly common protein expression workflow. And this may not exactly mirror your own workflow, but it's fairly common to start with some sort of cloning or gene synthesis step and then move on into protein expression, followed by purification. And then all of those steps are really just intended to get you down to this end of the workflow, down to characterization and analysis. And that's where you start the next phase of your project, right? So the faster that you get here, the better off you are. You have active soluble protein to work with, and you can start the next phase. But how many times have you gotten here and then realized that your protein is truncated or mutated, or you don't have enough yield to move on, or maybe your protein is even inactive? So that could be an issue, and you could end up having to move backwards along your timeline, right? Instead of being able to move forward, you're now moving backwards. And so you may go back to purification, you may have to go back to expression, or you may have to go all the way back and reclone your gene, which is really not fun. So what we'd like to do is be able to move forward always. And even along the way, sometimes we see some of these problems that might not even get you as far as characterization, right? So low expression levels, low solubility levels, maybe occluded purification tags or false solubility. All of these might mean that you have to work backwards in your workflow instead of forward. And that is, that repetition, it's costly in terms of time and money, okay? So we all know this. And just because there are so many places in the process to go awry, many of us decide to test multiple parameters in parallel in order to try to increase the probability of successful recombinant protein expression. So trial and error is very consuming, and if you can try a couple things in parallel and save time up front, it's a little bit easier, right? So this is just an example of what that might look like. Let's say you've discovered two new
looks like we may have had some technical difficulties with Beth's microphone. So basically, she's just describing an experiment where it's multiple parameters, and you have 2 sequences and 2 promoters and 4 tags and 2 strains when you're trying to evaluate proper expression conditions. So it can rack up to be around 32 samples. And this does not include the different samples, for with and without induction, your total and soluble and insoluble parameters or samples, and other parameters. So really the message here is that the experiments can get big very quickly. How can protein expression really go wrong, and why? So we have isolated the issue down to 3 major categories: low or no expression, inclusion body formation, inactive protein. So some of the possible explanations then for low or no expression is it's toxic, or you have rare codons or degradation. That's why you don't see any expression or very low expression. And based on our last webinar, we've identified the low or no expression as being the most problematic. And so we're going to focus mainly on this issue. So potential solutions are listed there: express protein in an active state and refold it, or you can use strains that supply limited tRNAs. Suppress the basal induction, then tightly control the induction level. So there are various tools that are available to you, and we'll discuss some of these more specifically. So the tunable promoters, we have a Rhamnose promoter that we'll talk about. You can try different engineered host strains. N-terminal fusion tags also help by getting the protein started. And so these are just some things to consider. I won't go through the entire table, but these are just some of the issues and some of the possible explanations and the solutions and tools.
Thank you, Karen. Sorry, everyone, I dropped out there for a minute. But thank you for continuing, Karen. That was wonderful.
No problem.
So yes, the items over here that are listed in red are really highlighted because they help to solve multiple problems, right? So the N-terminal fusion tags could help with inclusion body formations and low solubility, but they could also help with low expression or toxic expression. And they also may help with the N-terminal fusion tags, may help with inactive proteins. Same with tunable promoters. They show up in multiple places for multiple different problems. So if you're choosing strategies to implement in your process, these ones in red may be the ones to look closely at because they have the potential to solve multiple problems. And that will actually bring us to our first audience poll. So I'm going to hand it right back to Karen to give you guys the poll. Thanks for your participation.
All right, thank you. So here's our first poll question. What strategies do you use to express a functional protein in E. coli? Let me just pull that up, and here we go. So there's a number of different options here. Try expressing at different temperatures, different expression media. How about different tags? Optimizing the codon usage for E. coli. So please select all of the options that you've tried, or what's your strategy here. Maybe just give up and go home. Protein expression can be very difficult, as we've all probably experienced. All right, looks like we're finalizing the entries here. Try expressing at different temperatures. Seems to be quite popular. Well, thank you, everybody.
Okay, yes, thank you, everybody. That was interesting. It was very interesting that nobody picked go home, give up and go home. I'm very pleased to hear that. It sounds like you guys are a real hardworking crew. You really want to power through your protein expression experiments. So let's hope we can help you. So now that we've gone over what you guys like to use as strategies, maybe we can talk about some additional tools and strategies that we've developed here that can help you with those challenges. So the tool I'm going to focus on today is the Expresso Solubility and Expression Screening System, because it does help with many of these challenges on the left that we mentioned before. And it employs a lot of the solutions here on the right, and it's all wrapped up in one system. So we talked about low solubility before, and how adding a soluble N-terminal fusion tag can help rescue an insoluble protein. It can also help with low expression levels if the low expression levels are due to rare codon usage. So if the sequence of interest uses a lot of really rare codons, translation can stall out at the 5' end before it even really gets started. And adding an easily translatable fusion at the 5' end can help overcome that stalling effect. If the protein or mRNA is becoming degraded, an N-terminal fusion tag can also help. And in this case, it helps to stabilize that protein and prevent degradation. Tight promoters can also help with low expression levels or toxicity by controlling that basal level of expression and allowing us to fine-tune the expression. And then to avoid mutations, we use a Rec-A- strain, so recombination minus, for both cloning and expression. So the whole system is really designed to help you hedge your bets right from the beginning of your experiment. So you're giving yourself the highest probability of success in your initial screens for soluble protein. So the system starts with the beginning of experiment, which is amplification. The system includes a high-fidelity polymerase for amplifying the target sequences. You can certainly use your own high-fi polymerase if you have a favorite. We would just recommend that it be a proofreading polymerase. And we then have a panel of 7 ready-to-use vectors, each with a different N-terminal solubility and expression enhancing tag. And they contain a highly tunable Rhamnose promoter that drives gene expression. And we'll talk more about these vectors in a minute. We also include a Rec-A- strain of E. coli. And in this system, the same strain is used for both cloning and expression. And then there's a protease for removal of the fusion tag. And that also includes a control insert to make sure everything from cloning through protein expression and tag cleavage is working according to the way it should. Primers for colony screening and for sequencing your insert, and then reagents for induction of protein expression. So let's look at how you can increase your probability of successful recombinant protein expression using this system. You can use the system right from the start with your new or novel targets, or you can use it with targets that you are currently struggling to express. Maybe you've already tried a few things, haven't had as much success as you would like. You can certainly work this sort of system into your workflow. You can screen 7 different fusion partners to increase your chances of soluble protein production. And it's scalable, so it can be adapted to liquid handling instruments or to multi-channel pipette workflows. The system is also designed to save you time. So you design one set of PCR primers, and they're universal primers for your target. And you can use that amplicon to clone your gene into all those 7 vectors and test all those tags. And since the same strain is used for cloning and expression, it saves you that day and night incubation where normally you would need to transfer the expression clone from the cloning strain into the expression strain. So you can use the vectors for cloning right out of the box. They're processed for you, pre-processed, and ready to clone. And all of the cloning step itself is our Expresso cloning technology, which is faster than cloning with enzymes, and it doesn't require you to even do a PCR cleanup step. So the system is really designed to be a first pass at your target, so you can look quickly in only a few days at what fusion tags and expression conditions may hold promise for your targets. But the system is flexible and allows you to optimize if you want to. So if you would like to follow your initial expression experiments with some more fine-tuning, you can easily do that, and we'll talk about that too. So let's look at the workflow. You can test all of these fusion tags in the kit in a very short amount of time against your target or targets. And this is how it works. So we design one set of PCR primers for each gene of interest, not for each vector, but for each gene of interest. On the first day, we amplify the target and check it on a gel. And you're going to do a PCR product and an Expresso vector addition to the competent cells. So you just thaw the cells on ice, add your PCR product, add your Expresso vector, and transform E. coli. On day 2, we PCR screen the colonies and start overnight cultures. And if you take advantage of the hands-free autoinduction protocol, you can look at expression and solubility on day 3 and determine which fusion tags work for your protein. And then you can do a small-scale purification and tag removal for the tags you want to move forward with, and then move on to scaling up your experiments. So the key to what you can do with the system is really here in the design of the p-sol vectors. And this cartoon illustrates that although each vector has a different fusion tag, which we'll talk about on the next slide, the design of the vectors is actually universal. That's what allows you to design one set of PCR primers and clone that product into all 7 of these vectors. So they're small vectors. They're about 2.3 kb without the fusion tag. So we've removed a lot of that extra plasmid sequence that really doesn't perform any function, and it gives you plenty of room to clone up to at least 6 kb of your insert sequence. The Rhamnose promoter is inducible with virtually no leaky expression and very tight control. There's also an 6-his tag, an N-terminal 6-his tag, right, for purification. One of each of the 7 fusion tags that allow enhanced expression and or solubility, followed by a TEV cleavage site. The vector also has kanamycin resistance, which reduces the chance of satellite colonies that you sometimes see with ampicillin resistance plasmids. And the vector is also low copy number for increased stability, which really helps if you have a toxic or a difficult sequence. So let's look at the tags in the kit. 3 of them, this is a list of them. There's 7 tags and then an 8th vector that only has a 6-his tag on it, no solubility or expression enhancing tag. And this is your control, this is your baseline. And the rest of the 7 are all listed here, their full description. The top 3, AFV, SLY-D, and TSF, are novel tags. And then the remaining tags that are listed on the bottom here, these other 4, SUMO, BLA, MBP, and GST, are not novel. They are known in the literature and well used by many researchers. You'll also see that the panel of tags encompasses a wide array of sizes and isoelectric points, which may work well with your targets. And the 8th vector, that control vector, is only there again for a control. This is your baseline. So these tags weren't actually chosen at random, but in a very selective process based on a lot of experimental evidence, and I'll show you some of that today. But if you are experiencing any of those expression challenges we talked about earlier, this really is the only kit that offers you a panel of fusion tags to choose from. You may have a couple of these already hanging around in your freezer in your lab, but you probably can't use a single PCR product to clone into them. So you lose that ability to parallel process from cloning all the way through your expression screens. And this system does allow you to do that. And this will bring us to our second audience poll. So I will shoot you all back over to Karen for the poll. Thank you.
Thank you, Beth. All right, here's our second poll. And which solubility expression enhancing fusion tags have you found to be most effective in E. coli? Again, choose all that apply. There's some very popular ones up there, GST, MBP. Some that you may not recognize, and we're curious to know if you utilize those as well. And then 7 and 8, and 7's none. I use fusion tags for detection purification, but not for solubility expression. That may be a common like his tag option. Or you don't use fusion tags at all. All right. Looks like MBP is a favorite. Or none, just use it for detection purification. Great. Thank you, everybody.
Okay, Beth. Okay, thank you, everyone. That's good to see. So now we'll take a little bit of a closer look at the protocol and some data and results. So the first step, protocol day 1, is to amplify your gene of interest. And we're going to add 18 base pair Expresso overhangs to our PCR primers. So this is a little cartoon showing the map of the insert area. So this is the Rhamnose promoter up here. This is a sequencing primer, a landing site. Then there's a 6-his tag followed by a fusion tag of some sort, right, one of the 7. And that is then followed by the TEV cleavage site. Then this is where your target gene will be inserted, followed by a stop codon, and then some more vector sequence, and then a reverse sequencing primer is here. So because of the universal design of the vector, again, adding these 18 base pairs onto the 5' end of your primer, of your forward primer, and these 18 onto the 5' end of your reverse primer is going to allow you to clone into all of those 8 vectors simultaneously. So then we're going to check our PCR product on a gel, and if the PCR reaction yields that single, robust, nice, juicy band, there's no need to clean up your PCR product. All you need to do is thaw your competent cells on ice and add 2 microliters of your PCR product, 2 microliters of your P-sol vector, and proceed with transformation. So it really is that easy. The cloning in Expresso cloning is mediated by homologous recombination, which actually happens naturally inside E. coli cells. So it's fairly simple and straightforward. And you may be wondering if it works because there are no incubation steps with an enzyme. But in fact, it's a very high efficiency cloning method. Here are the results of 2 colony screens, which is something that you could do on the next day, on day 2. These are both Expresso cloning experiments. In one case, 17 out of the 18 colonies picked were correct by PCR. And in this case, we were looking at a number of different fusion tags and picked 2 colonies from each plate, and all of them were correct, 24 out of 24. And this kind of result with Expresso cloning is not unusual at all. We normally see greater than 95% efficiency in the cloning steps. And honestly, between you and me, you probably could skip this PCR cloning step or PCR screening step unless you have a very large gene or something that you know is just unstable in the background of the cell. So in that case, we would expect that you would want to do a PCR screen to make sure that you've got full-length product cloned in. But if it's not challenging, then you probably will be okay doing a lack of PCR screen. But either way, the result is that you'll start cultures overnight from your positives. Now, we'll talk about induction of protein expression, and you can do this in a couple of different ways. So the standard induction is to start an overnight culture from your colony and harvest part of that culture for mini prep DNA and sequencing, and then if you would like to prepare a glycerol stock, you can do that as well. And then use the remaining, or part of the remaining culture to, as a starter culture to inoculate fresh medium for induction of protein expression. And so if you inoculate 3 mils, then we let that grow to an OD of about 0.5, and then add our inducing reagent, which is Rhamnose, and harvest after about 4 hours. And as you can see here on the right, this gel shows the response of protein induction to different levels of Rhamnose. And we start out very small. We start out with 0.001% and go up to 0.2% in a gradient. And you can really see that the level of protein expression is highly responsive and relative to the amount of Rhamnose that is added to the induction medium. This line here is LB only without Rhamnose. So there's maybe a hint of something going on there, but really, I mean, virtually no expression going on. And then this lane is actually no Rhamnose, but with 2% glucose. And glucose acts as a repressor for this particular system. So there's very tight control. There's absolutely nothing coming through there that we can see on this gel. So if you have a very difficult or toxic protein, you can always back down on your Rhamnose concentration and slow down your expression levels, slow down what your cells are doing, and try and get that toxic protein to be expressed at a level that the cell can tolerate. So now if we look at the other way that we can express proteins, it's via autoinduction. And this is where we start to culture from a colony, and we add both glucose and Rhamnose to the media at the same time, just when we're starting the colony. And then we walk away and we let it go overnight. And the cells will preferentially grow up on glucose. They'll utilize glucose over Rhamnose and build up biomass until the glucose is depleted from the media. Then they will switch and start metabolizing Rhamnose. And at that point, and only at that point, the Rhamnose promoter will be activated and expression will be induced. So you can control the timing of the induction by how much glucose you add to the media. If you add more glucose here, so for an early induction or a late induction, right, we would add more glucose for a late induction, where the cells would grow longer on glucose and then induction would show up over here around 24 hours in this example. If we wanted an earlier induction, we would add less glucose. The cells would build up less biomass, but we'd get our induction started a little earlier. And this also can really help with the expression of toxic targets that are deleterious to the cell upon induction, because the regulation on this promoter is so tight and there's virtually no leak, as you saw in the previous slide. You can build up a substantial cell biomass on glucose before the induction and accompanying cell toxicity begins. So more biomass means that even if you only get a few hours of expression before you see the toxic effects on your cultures, you have a better chance of getting more protein out of the culture. And this is also in contrast to the results that you can get from a T7 promoter, which is a very strong promoter, but it's really either off or on. It's really hard to fine-tune it the way you can with the Rhamnose promoter. So So let's look at a literature example where Rhamnose promoter was used to express a particularly large and difficult protein called lymphostatin A, or lifA. It's an E. coli virulence factor, and it hangs out. It's an enteropathogenic E. coli. So it's very large. It's about 365 kilodaltons. It's one of the largest bacterial toxins known. And there are previous literature references that indicate it is very unstable in its recombinant form. They were able to clone it from they were able to clone it into a cosmid or phosmid library, but they were not able to express it recombinantly. However, this team from the Roslin Institute in the UK, down here in references 3 and 4, were able to successfully clone and express it in the Rhamnose promoter system. And they took advantage of glucose repression. They started their cell growth at 37, grew to a low OD, and then backed it down to 30 degrees and did a short induction with a lot of Rhamnose. And that worked for them. And they were successful at this, and then they went on to generate some functional mutants of this protein. They were able to generate those mutants via site-directed mutagenesis and characterize them after expression. And then just this year, they went even further and cloned and expressed a homolog in the same system. And in this case, they induced, again, with 2.2% Rhamnose, but they did a cold induction at a much longer time frame. Both of these proteins in their native state have anti-proliferation effects on T cells, and the authors were actually able to prove that the recombinant forms have the same inhibitory effects. So this is just one example of the Rhamnose promoter's ability to express very toxic proteins and very large proteins. This is a page gel. Both of these are page gels and Western blots of purified protein, and you can see it's a very large size protein. So now, let's look at the next step, which is evaluating protein solubility and expression levels. This is the point we all want to get to, right? Here's the data. So what we normally do, and it depends on if you do the standard induction or the autoinduction protocol. If you use the one-night autoinduction protocol, you're doing this on day 3. If you do a standard induction where you grow a starter culture and then start an overnight culture from there to induce, then you're doing this on day 4. Just depends on where you are and how you want to do your experiment. But we grow E. coli clones and induce expression with the protocol that you would like to use. You can take a sample of the uninduced cell culture if you would like to. Harvest the cells by centrifugation and resuspend them in sonication buffer. And at this point, if you're planning on purifying this little sample, you probably want to keep the purification requirements in mind. So DTT, protease inhibitors, just think about those things while you're getting your sonication buffer together. And then lyse the cells by sonication, and then take a sample of total protein. So that gets loaded on your gel over here under T. So for each fusion, you're going to generate 3 samples, and the first of which is going to be the T, or total sample, total protein sample. Then we fractionate the lysate by centrifugation and take a sample of the soluble protein from the supernatant. So that goes here next door in the S lane, right? So you've got total protein and then your soluble protein, and then resuspending the insoluble pellet in sample buffer and boiling it gives you the insoluble fraction. And then you analyze all these 3 fractions for each tag on an SDS page gel. We usually do it in 2 15-well gels, and it works really well. Then we compare solubility and expression to the control. And this is where the control vector, that control construct really comes in handy, because this is your baseline. This is without any solubility or expression enhancing tags. This is your baseline where you are. This is with a p-sol fusion. And you'll note they are 2 different molecular weights, but that's because this protein has a p-sol fusion on it, right? So it's going to be a little larger. But we're looking for an increase in the level of soluble protein, which we see here. That'll tell us we have an increase in solubility, and an increase in total protein in the T lane. That'll tell us we have an increase in expression. So we look at all of our p-sol fusions in comparison to the 6-his control, and then we determine which p-sol fusion clone or clones to proceed with tag removal and characterization and other things that you would like to do with your protein. Scale up, whatever it is you would like to do. So let's go through a couple of examples of data. And in the first example, we're looking at GH1, which is human growth hormone, and it was expressed with all 7 targets plus the control. And the expression levels and solubility levels were evaluated just as I showed you. And they're all different molecular weights, again, because the tags are different sizes. So here is a 6-his baseline control. And in all of the cases highlighted in green, these tags were able to assist with both expression and solubility levels. Some of these other tags, let's see, like SUMO, that appeared to enhance solubility, but it didn't really help all that much with expression, for example. So that's not necessarily one we would move forward with, especially when we have great examples in BLA, MBP, and TSF. Those are the 3 we would probably move forward with for sure. Now in our next example, this looks a little different because it's a different protein. And different tags tend to rescue different proteins. And this is something we found consistently throughout the work that we've done here. In this example, fewer tags were successful at all as well. So the fusions in green were able to help both solubility and expression, but really it's not as dramatic as effect as you saw in the previous gel. And the tags in red helped either solubility or expression, but not both. And this is something that you want to keep in mind too. The AFV and SLY-D samples actually have a co-migrating E. coli band that migrates just where the fusion band is. So it's pretty easy to see some of the expression results because that band does appear a little bit stronger than it does over here in the 6-his control. But the solubility results are really kind of hard to read. We don't know what proportion of that band is actually due to the co-migrating band and what part might be due to increase in solubility. So in this case, we would recommend moving forward with TSF or MBP first and only coming back to AFV and SLY-D if absolutely needed. Or you could always, clean this up a little bit, maybe even run it over a column and rerun it to really look at the yield here. So different proteins or different tags rescue different proteins. And in our next example, we have a restriction enzyme. And in this case, the tags rescued one parameter, but not the other. So there were a few tags that improved expression here in the T lane, but not really any of the tags improved solubility at the standard conditions that we were using. So in this case, we'd recommend doing some expression optimization on the tags that showed promise for enhanced expression. So lowering the growth temperature, reducing the Rhamnose concentration. It may be possible that there's a trade-off here between expression and solubility. And if that's the case, then the Rhamnose promoter is actually a great tool for you to finely tune that expression level, to find just the right balance between expression and solubility, and still keep your cells alive. So the next example I'll show is a DNA modification enzyme. And this is a case where we were able to help use the panel to identify a common problem. So you'll see over here in the his lanes, right, our total, soluble, insoluble, we see 2 bands in our insoluble protein samples. And we see it here as well with GST. We see it again with MBP. In every case here where we see a protein band, we also see another band just slightly larger. And that larger band is actually the correct molecular weight. So what we think we're seeing is a fast degradation of that product upon expression or upon lysis. We're not, we weren't sure which. But this is a case where we would probably recommend moving forward with one of these green boxed tags, so either the TSF and the MBP, and maybe making sure that we add protease inhibitors, right, because it could be protease digestion that's happening here. It could be proteolysis, to all of our sonication experiments, and even maybe moving this construct into a protease deficient strain for expression. But because we were able to see this result with all of these tags, it made it pretty clear that it was some sort of degradation or shorter product that was being transcribed or translated somewhere along the line or degraded. if you saw this with one tag, you might think, oh, well, I think this is a degradation product, but I'm not really sure. Maybe I should run a Western. Maybe I should clean it up and see if it sticks to a column. In this experiment, since we're seeing the same thing across all of these tags, it really makes it fairly easy to identify what's going on. So I mentioned that these tags were selected from a larger panel by trial and error. What we did was to mine the literature for targets that are known to be challenging to express, and then we cloned and expressed them. And some of those targets and some of those tags that we found in the literature as well just naturally dropped out. So some of the targets that were supposed to be really difficult didn't turn out to be so difficult in our hands. And some of those tags that we tried were really not good at either solubility or expression enhancements. And this is just a sliver of that data where we ended up taking 12 of those potential tags and matrixing them against 8 different targets that we had found to be truly recalcitrant in either solubility or expression. And the graph here illustrates how many of these 8 proteins each tag was able to rescue. And the yellow bars indicate solubility rescue, and the red bars indicate expression rescue. So in the case of AFV, for example, AFV rescued 3 of the 8 for solubility and 3 of the 8 for expression. TSF, by contrast, rescued 5 of the 8 for solubility and 4 of the 8 for expression. So this is just an example of the way that we collected some data, and there were some clear winners and losers in our panels that enabled us to winnow down the results. And another piece of data from our experiments here is that all tags are not created equal. We definitely were able to confirm this. Some of them are definitely more adept at fixing specific problems than others. So out of the 24 plus targets we tested internally, some of them were better at fixing things like codon bias. Some of them seem to be better at fixing problems like low expression, and some of them seem to be more adept at fixing solubility problems. This is just a peek into some of the data that we generated and what we found. We would expect that, with your own targets, that'll probably be very different. But this is just to illustrate that we were able to see some trends, at least in the kinds of proteins that we were working with. So let's talk about scale-up. This particular system does offer many options for cloning and transformation. The component cells are really one of the key components that enable scale. So the kit is provided with chemically competent cells in tubes, so 1 transformation per tube. Very, normal transformation like you're very used to doing. Again, except that you're doing your cloning and your transformation basically in the same step. So you thaw cells, add the Expresso pre-sol vector, and then add the PCR product and just transform using a standard protocol and plate. If you're doing multiple samples at once, the protocol can be easily adapted to use our 10G cells in 96 well plates. Same cell line, different format. But it's amazing how a 96 well plate formats itself very well to 8 different vectors and then multiple targets going across. And these are actually tearaway plates, so they can be broken off into groups of 24. So if you're working with like 3 targets, you would just use this portion of the plate, and the rest of this you wouldn't even have to thaw. You can just tear this off and go. And then after that, these transformations can be plated on a Q tray or other sorts of plating media, whatever works for you. And then you can use 96 well deep well plates in order to do the expression experiments and the induction experiments. So it's all very amenable to high throughput multi-channel pipettes and or liquid handlers. And we can help with that as well. So here's an example of the scalability. This is functional annotation of a gene. This is functional annotation of a bunch of genes, actually. So it's an organism called Fibrobacter cystogenes, and it's a rumen bacterium, and it helps cattle digest cellulose. So the whole genome sequencing revealed that there was a subset of 48 enzymes that looked interesting, that they wanted to do some expression experiments for. So they did a 96-well PCR amplification and cloning step, right, into the Expresso vector, and 96-well expression induction, 96-well assays, right, on the induced sample lysates. And that's one thing we would recommend as well is if you have a protocol where you have a good assay for your protein, try and work that in as quickly as possible into your workflow so that you're not going down the wrong road. You want to find out if you've got functional protein as quickly as possible, right? So in addition to doing those 48 enzymes of interest, they also did a shotgun expression library in an Expresso vector, and they screened that for enzyme activity. So all in all, over 400 proteins were produced in the system. And here are a couple of references that you can check out should you want to do so. So if you get the system and you decide, oh, I have tested this system with multiple different targets, and I find that there are just a couple that seem to work repeatedly for me for whatever reason, we can work with you. We can provide those tags that work for you at larger scales if desired. We do this as a custom project. If you find there's some vectors that you don't want to use the whole panel of 8, that's fine. We'll work with you on a custom basis to provide it in the formats that work for your workflow. So just another option for you. And let's go over some quick tips for success here. So in the Expresso cloning workflow, remember these T-cell vectors are all kanamycin resistant. And because we don't necessarily need to gel purify the PCR product before cloning, if that gene to be amplified is carried on a kanamycin-resistant plasmid already, if that's your PCR template, you might experience some background from the transformation step. So we need to kind of get rid of that template, that template DNA, kanamycin-resistant DNA. So a couple different ways we can do that. We can gel purify the PCR product before we clone it. We can also just treat the PCR product with DPN1. That should chew up any template DNA that was generated in a normal E. coli strain. And then we could also just linearize the plasmid template with a restriction enzyme before performing PCR. We also recommend using chemically competent cells. Expresso cloning is actually not compatible with electrocompetent cells. So we definitely want to use the chemically competent format of our cells. And then to maximize your transformation efficiency, especially when you're doing the tube protocol, we found it necessary to transfer the cells and DNA to 15 ml polypropylene culture tubes right before the heat shock to make sure that we get optimal heat transfer. For the expression screening part of the protocol, we'd recommend measuring the growth rate of your transformed strain before and after induction, especially if you're trying to optimize or if you see that there may be something, if you're seeing some cell death or any toxicity. And you want to probably measure it against the growth rate of the parental untransformed strain. And if the growth rate changes significantly after induction, you may be starting to see some toxicity. And if you do see that, we would recommend trying a gradient of Rhamnose concentrations. That's one thing you can do. And it's fairly straightforward. You could also perform an autoinduction with increased amounts of glucose, like we talked about before, to increase that cell density prior to the point of induction. And then you could also try, standard lower growth temperatures. Just note that slowing down the cell growth rate may actually require longer induction protocols in the end. But if it gets you soluble protein, hey, that's awesome. If the protein looks to be insoluble, though, some of the same mitigations actually, lowering the growth temperature and decreasing Rhamnose concentration. You can go as low as 0.001%, as we saw before. And it gives you a very small amount of protein, but it might just be soluble protein. Assay your protein as early in the process as you can to find out if you're on the right track. And then another note here, unlike T7 promoters, the Rhamnose promoter is actually active and inducible in almost any E. coli strain. So clones can be transferred to other favorite strains, so for expression studies. The Rhamnose promoter does not at all depend on the phage T7 RNA polymerase. So it only depends upon the E. coli RNA polymerase, which is present in almost every E. coli strain unless it's been engineered out. And we have not found a strain yet that's the case. So the promoter is inducible in all strains. And so if you would like to use a protease-resistant strain to do your expression experiments in, you certainly could. So to summarize, our Expresso solubility and expression screening system really is meant to be a quick overview of different options in terms of expression and solubility tags and expression conditions that can help get you on the right track with your new targets or targets that you're struggling with. It helps to increase expression and solubility. And it also has a very simplified workflow. We've worked hard to make sure that it tries to save you time and simplifies what you're trying to do. Because let's face it, as in our first poll question, protein expression is hard. Whatever we can do to make it easier, we want to try to do that. We also tried to do what we could to facilitate downstream analysis and characterization by making the tags cleavable, which is the subject of the next portion of our webinar. But first, we do want to mention that we have flexible kit conformations for this as well. So these are 4 different catalog numbers for this system. And the PSOL vector suite and the E. coli 10G cells and tubes are included in all of these. However, SelecTEV Protease, that we'll talk about a little bit here in a minute, and the Accura polymerase are 2 things that are sort of optional. So you can get a kit that includes those if you like. If you don't like to, you can order without. And the SelecTEV and Accura are also available separately. If you do not have a hi-fi enzyme that you want to use or love to use, we would like to recommend a new system to us. This is a system that came to us through our distribution of Epicenter products. This is the fail-safe PCR system. And for those that are not familiar with it, the slogan that goes with this particular PCR system is never fail at PCR again. So it has 12 2x hi-fi PCR premixes containing buffer, magnesium, PCR enhancer, and DNTPs. And you perform your PCR reactions with your target in all 12 premixes to try and find the premix that amplifies your particular target the best. And then if you find that particular target amplifies with, say, in this case, buffer J, buffer J is available separately. So you can just order buffer J next time. If you're doing a lot of amplifications of this particular target, you can just use buffer J next time and order more of that. So it's a very flexible system, and it's great for new targets, things that you haven't amplified before. You're not sure exactly what amplification conditions to try. This gives you a lot of great options. So protease tag cleavage, onto the last portion of our webinar here. And that'll bring us to our 3rd and final audience poll. So I'll hand it back to Karen for that.
All right, thank you, Beth. Which protease do you prefer when removing a fusion tag from your protein of interest? Again, please select all the options that apply. TEV, SUMO, thrombin, enterokinase. Which protease do you prefer? Maybe you don't need to remove the fusion tag, and so you don't have any preference. Thrombin? Wonderful. Okay, it looks like TEV is one of the more common ones, or several don't need to remove my fusion tag. All right. Thank you.
Okay, great. Well, it looks like there's a number of you that definitely don't need to remove your fusion tags, and that certainly does make life easier. For those of you that do have applications that you need to remove them for, this next section may be interesting to you. So these are sort of our rules of thumb for when fusion partners should be removed, and it really is all about your downstream application in our view. So it's hard to predict which proteins will retain their structure and function with a fusion partner. Or really for an application such as like crystallography, large fusion partner removal is usually recommended, but peptide tag removal, it may not be necessary. There are plenty of proteins that are successfully crystallized with tags on them, and even fusion partners. So These aren't hard and fast rules, right? For enzymology, bacterial or biochemical or other activity assays, we probably want to try and remove those fusions whenever possible. But again, assay the protein first. And if it's active in your application, tag removal may not be necessary. And then for large-scale production or therapeutic use, I mean, these are just examples, right? Tag removal is often recommended if not required. And then thinking about where your tags might be on your protein, it kind of depends on the intended use of your fusion as to where you would like your tags. So solubility or expression enhancements, if that's the intended use, that'll probably be on the N-terminus to get the protein really that good running start during translation. If the fusion must be removed, usually that is also at the N-terminus because the proteases tend to leave more amino acids if they are a C-terminal fusion than an N-terminal fusion, just the way a lot of them are designed and the way that a lot of them cleave. And then if you have a tag for affinity, purification, or detection, that could be on the N or the C-terminus. And this is not intended to be an exhaustive list, but it's some of those proteases that we just mentioned in our quiz, poll. It wasn't a quiz. It was a poll. EK, Thramin, Factor XA, Sumo, and TEV. And then, different molecular weights, different cleavage recognition sites. And as you see, different amounts of residual N-terminal amino acids left on the protein if it's an N-terminal fusion. And then what's really, they all have their pluses and minuses, but really here the specificity is a pretty big deal. So Factor XA and Thramin and EK really have, on the poorer end of specificity, whereas Sumo and TEV are definitely more specific. They are also work in a wide range of pH and salt and temperature conditions, and they're pretty well characterized. Some of these proteases do have limitations in their tolerance for different amino acids in the P1 prime position, which is the amino acid immediately after the cleavage site. So if it's a big amino acid with a large aromatic group, there can be steric hindrance there, which will prevent efficient tag cleavage. So just something to keep in mind. All right, so this is a workflow illustrating our SelecTEV and Sumo Express protease workflow for tag purification, or target purification rather, and tag removal. And both of these proteases are 6-his tagged. And if you're not familiar with using 6-his tagged proteases, it's a pretty slick way to remove the protease from your gene of interest or from your target of interest after cleavage. So first we express and purify the tagged target by IMAC on a nickel or cobalt column. So we have our purified tagged protein with a 6-his tag protease cleavage site. This is the actual site here in red, and then the target protein. And we add our 6-his tagged protease, either SelecTEV or Sumo Express, and incubate. And that will release the tag from our gene of interest, or our target of interest rather, leaving our target protein, untagged target protein. And so we have a mixture of our protease, the 6-his tagged tag, and our protein of interest. Then we do a subtractive IMAC step. So basically what that's going to do is it'll bind the 6-his tagged protease and the 6-his tagged residual tag, and the target protein will flow right through. And the target protein will actually be in the column flow through and the first wash, and that's where you recover it from. So it's kind of a slick way to remove these contaminating bits and get your purified protein back. So the 6-his tagged SelecTEV protease, again, this is tobacco etch virus protease. It recognizes the normal TEV recognition site with a glycine here in the P1 prime. This particular TEV protease is engineered for improved stability and activity, but it is active from pretty normal pH range with normal TEV as well. pH is 6 to 8.5. Optimal temperature at 30, but has a very wide range, and it tolerates some chaotropes there as well. So this gel on the bottom is cleavage and purification of GH1, which was the first example of expression with the solubility kit that we went over a few slides ago. So here in lane 1 and 2, we have before and after SelecTEV cleavage. So the top band here is the whole fusion protein with the 6-his tag and the fusion and GH1 all together. And then in the second lane after cleavage, you can see GH1 is down here, and then we have the 6-his blah tag here, and the SelecTEV protease is there as well. And then in lanes 3, 4, and 5, this is the subtractive IMAC step. So lane 3 is the flow through from the subtractive IMAC. It just shows just GH1 coming off the column in the flow through, and then a little bit here in the wash. It's kind of hard to see. But then in lane 5, this is the column elution, and this is where we just eluted what was left on the column with the imidazole and came away with the 6-his blah tag and the SelecTEV protease, which are both 6-his tagged. And then we have our GH1 is purified. And then our 6-his tag sumo express protease is actually optimized to cleave a stabilized sumo tag. So it's not the normal wild type sumo tag. This is a specifically engineered tag and a specifically engineered enzyme for cleavage of that tag. And the cool thing about the tag is that the sumo tag itself is resistant to eukaryotic desoymolation enzymes, which allow you to take this construct and clone it into a mammalian expression vector and express the protein as a sumo fusion protein in insect or mammalian cells. That does not work with regular sumo tags. It only works with this engineered sumo tag. So you can transfer your fusion to a mammalian system and still retain the benefits of the sumo tag, should you so wish to do so. It does exhibit reduced activity with the native sumo sequence, so it will cleave the native sumo tag. It's just not very efficient. And native sumo protease will not cleave this special sumo tag, this stabilized tag. But it has very similar reaction conditions to normal sumo protease. Active in a wide range of pH, optimal at pH 8. Again, 30 degree optimal, but active from 4 to 37. Fairly decent salt tolerance, not quite as good as TEV, and then non-ionic detergents are also okay at 1%, and it tolerates some K-tropes as well. So this is a similar gel to what I showed you in the last slide for TEV protease. So this is without and with sumo protease. So before cleavage, we have the whole protein up here. After cleavage, we have our sumo express protease, and we have our release tag and also our gene of interest. And then here in the P, this is the purified target protein after subtractive IMAC, and that's what we have for purified protein after the IMAC column. Okay, so best practices. When possible, we would advise you to choose a protease with a molecular weight that's significantly different from your protein. That way you can easily check for tag removal by page gel analysis. We also would recommend using proteases at the recommended concentration first and then optimize if needed, because if you go much higher, it can lead to nonspecific cleavage. So you want to watch the time and the amount of protease added fairly carefully. And remove various samples at different time points and analyze them by SDS page when you're optimizing. We would also recommend that you keep in mind the 6-his tagged fragments and the protease 6-his tag protease itself when you're calculating column capacity for that subtractive IMAC step, because those proteins will bind to the resin, and you want to make sure that they're both are accounted for, and you're not exceeding your column capacity and flushing some of this through in your wash with your protein of interest and in your load. DTT can also cause leaching from some nickel resins. If you haven't experienced this, it is not very fun. So we recommend testing your IMAC resin before putting your protein on it, just to ensure compatibility with the DTT concentrations in the recommended protease reaction buffers. It's 1 or 2 millimolar DTT, so it's fairly high. And if needed, if it's not compatible, you can always dilute your sample before loading to reduce that DTT concentration going on your column. If you do see incomplete tag cleavage, we would recommend, again, adding more enzyme and maybe incubating for a longer time period. You could even bump it down to 4 degrees and let it go overnight. And then dialyze your protein if needed to remove those potential inhibitors, so interfering detergents or imidazole could be interfering with some of your tag cleavage. And then if you see false solubility, this is not fun. This is when you remove the tag with the protease and all of a sudden your protein crashes out, so it's just precipitated. It tends to be more common with larger proteins or proteins with very large hydrophobic patches, right, the very difficult ones. And what we would recommend is to test other fusion constructs that exhibit apparent solubility and try and vary that pH and salt conditions during purification and after tag cleavage. And maybe use a buffer that differs from the PI by 0.5 pH units. Just don't go below pH 6 if you're before a nickel column step, because that will cause your protein to not bind to the nickel column. But false solubility is definitely a challenging situation. This is just a quick guide for our different Expresso protein expression systems. And it starts with what your application requirements are. So your downstream application, is it okay to have one additional glycine residue on the N-terminus? If so, then the solubility and expression screening system is okay, because by the time you cleave with your SelecTEV protease, it will leave one additional glycine residue on the N-terminus of your protein. If a 6-his tag is acceptable and you don't need to cleave it, you could use the T7 system or the Rhamnose system. This will also tell you what fusion partners are available, what termini they are on, whether or not tag removal is possible with that system, and then whether or not the protease is included in the kit. So it's just intended to be a reference guide for you based on whatever your application and your requirements are. So here are a couple of resources that you may find useful. We may send some of these in a follow-up email to you after the webinar as well. Hopefully you will be able to check them out. We will also make the slides and the recording available to you. You should be getting an email when those are posted. And with that, that's all the material I had.
Thank you for your participation in the webinar, everybody. If you again, I want to mention both the recorded webinar and the slides will be available on our website in a few days. I want to thank Beth once again, and thank you, everyone. The webinar has now concluded. Thank you.
Thank
Have a good day.
Karen. Thank you to all your listeners.